And then a hash value is separated into two parts with Q bits and R bits respectively, called quotient and remainder. The quotient is used to locate the slot in CQF, and then the remainder will be inserted or the corresponding counter C will be updated, depending on whether this remainder has already been inserted or not. CQF guarantees zero false negatives but has false positives due to hash collision. The false positive rate of CQF can be computed by this formula. Our proposed k-mer counting method, CQF denoise, implemented CQF with several advancements. Among them, the most important one is a denoise module to remove potential false k-mers on the fly. Our denoise module can determine the suitable time and the number of rounds of noise removal based on a user-specified tolerable rate of true k-mers being wrongly removed. And we determined that rate by some probability calculations. How we determine the suitable time for denoise is demonstrated in this flowchart. Given the sequence scenario profile, we can estimate the true to false k-mer ratio by dynamic programming. And then based on true to false k-mer ratio, number of k-mers in total, and number of unique true k-mers, we can compute the mean counts of true k-mers. By assuming that the counts of true k-mer follow a Poisson distribution, we can estimate the number of denoise rounds in such that the probability for a true k-mer to occur in or fewer times is less than the tolerable round removal rate of true k-mers. The number of false k-mers is computed based on the true to false k-mer ratio and the number of k-mers in total. Our goal is to remove all false k-mers in n rounds of denoise evenly in order to minimize the size of CQF. To do that, the denoise is triggered when number of unique k-mers in CQF reach a certain threshold, which is the number of unique true k-mers plus the number of false k-mers divided by the number of denoise rounds. In our experiments, we use 5 data sets from 4 species with different genome sizes and sequencing depths. Using those data sets, we evaluated the effect of denoise on counting efficiency and accuracy. And we also evaluated the counting and query performance compared with existing methods. And we investigated the scalability on sequencing depths. We used the C. elegans dataset to evaluate the effect of denoise since it has the highest sequencing depths. Panel A shows that the frequency distribution of k-mers in C. elegans datasets is multimodal. It has a peak at around 160, which likely corresponds to the median coverage of true k-mers, and another peak at 1, which should contain mostly false k-mers. And we observe a local minimum at around 50, and so we use it as a demarcation point to manually set the number of denoise rounds for different values from 0 to 50. Panel B shows that as the number of denoise rounds increased, more and more true k-mers were wrongly removed, but the round removal rate remains low, reaching a maximum of around 10 to the power of -24 with 50 rounds of denoise. At the same time, the memory usage dropped rapidly from 5.5 gigabytes to 0.9 gigabytes when the denoise rounds increased to 20 and is then stabilized. Those results suggest that denoise removes few true k-mers but reduces memory usage substantially. We also evaluated the effects of denoise on counting accuracy. We first studied the singletons, which are k-mers occurring only once in the read. Panel A shows that less than 0.2% of singletons were removed by denoise and retained in the CQF due to hash collision, and less than 2.5% of non-singletons were removed by denoise, as shown in panel B. And next, we directly compared the camera counts computed by our method and the actual counts. Panel C shows that less than 4% of the cameras had an incorrect count, and among them, more than half had a count of difference only 1. Together, those results suggest that the counting accuracy is minimally affected by the noise. And then we compared the counting performance of our method with others 4 state-of-the-art camera counting methods in all 5 data sets. As shown in panel A, our method was consistently the most efficient method in terms of the memory usage in all 5 data sets. And regarding the running time, CQF denoise was similar as the Jellyfish 2 and Swagger, and it was much faster than BF counter. It was not faster than ChemC3, but the short running time of ChemC3 came with the cost of a much higher memory consumption than our methods. Together, they show that CQF denoise uses 49 to 76% less memory, but runs as fast as memory-based methods. We also evaluated the scalability of those methods on sequencing depths. Panel A shows that CQF denoise consistently used the smallest amount of memory, and the increasing of the sequence depths had minimal effect on the memory consumption of CQF denoise. And that was not the case for the other methods. Regarding the running time, the order of different methods almost remain unchanged at different sequencing depths. Together, they suggest that the memory usage of CQF denoise is nearly unaffected by sequencing depths, and it consumes the least amount of memory but runs as fast as memory-based methods. We further developed a de novo deburring graph-based assembly method, SH assembly. Our method first counts the k-mers using CQF denoise, and then starting from k-mers sample from race, we extend the deburring graph by querying for the existence of next k-mers in the CQF. And there could be false positive k-mers in the deburring graph. And after constructing the deburring graph, we have a follow-up step to identify and remove short branches and other suspicious nodes. And after that, the contigs can be constructed from the deburring graph. We use our method and other 3 assemblers to assemble the sequencing data of C. elegans and Homo sapiens into contigs. For C. elegans data assembly, assembly consistently was the most efficient method in terms of both the memory usage and the running time. For Homo sapiens assembly, assembly used only 2.7 hours, and the memory usage of assembly was much lower than other memory-based methods. And regarding the assembly quality, assembly was consistently one of the best methods in terms of NG50 and GA50, total contig length, and total aligned contigs and genome coverage. And those results show that our assembly method, sorry for that. Our assembly method requires less time and usually less memory without affecting the assembly quality.
Okay, so.
To wrap up, we propose an efficient and robust camera counting method which uses CQF with a module to remove potential false cameras on the fly And our k-mer counting method uses less memory than other state-of-the-art methods, but runs as fast as other memory-based methods. As application of our k-mer counting method, we develop a de novo assembly method, and our assembly method gains significant runtime and memory improvement over existing method and achieve comparable results.
Okay.
And thank you for your attention. Yeah.
Thank you, Christina. Are there any questions? I don't see any in the panel. So I'll ask you one question already. When you're comparing the different assemblies, actually your assembly was not among the better ones if you consider misassemblies.
Yes.
So
Yes.
Can you explain this?
The number of the misassemblies of our method is actually similar to that of the mini-3. And that's most because we used the graph simplification module in the mini-3 for, after we're building the deburring graph, we need to simplify the graph and then deduce the context. Yeah, and because we use the graph simplification module in mini-3, and that's why the misassembly statistics are similar as that of the mini-3.
Okay, I have a question. So whether you could give a bit more details on how you actually do the denoising. How does it work?
The denoise procedure is triggered when a certain when the number of the total number of unique k-mers in the counting data structure reach a certain threshold, and we determine that threshold based on both the number of true k-mers and number of the false k-mers estimated in the sequencing data.
Short, maybe
for more
question?
details, I refer you to our paper. Yeah.
Okay. How do you deal with repeats? Like plant genomes have very large repeats, a lot of repeats, so the k-mer count might be biased.
Yes, for those k-mers in the repeat region, they will have much higher occurrences counts than those not in the repeat region. Yeah, so our assumption of the Poisson distribution may not applicable for those k-mers in the repeat region, and I think this is one of the problems that we want to handle in the future. And yeah.
Okay,
thanks.
Christina, thank you. So we'll have to move on to the next one.
Thank you.
Thank you. So the next one will be Maor Asif from the Ben Gurion University of Negev. So this is using a method to actually pinpoint transcription factor binding sites in a more efficient and accurate way. Okay. Maor, the floor is yours.
Okay, good morning. Pleasure to be here. Today we're going to produce DeepSelect, which is a method to infer DNA binding preferences from HT-Select data using multi-class CNNs. So let's get started. Let's first introduce gene expression regulation, which is a complex biological procedure. The gene expression regulation depends on transcription factor protein, which bind to DNA subsequences called binding sites. Those binding sites are located mainly in the gene's promoter near at the edge of the gene near its 5' end. Transcription factor can repress or promote genes, which mean to decrease or enhance them. Here we can see two transcription factors near the edge of the gene at its promoter. So high-to-output selects, or just in short, HT-Selects, it's an in vitro technology which is capable to produce several million high-resolution sequences in each experiment. The experiment starts with a very large pool of DNA sequences. Those sequences are exposed to a transcription factor protein. Sequences which were bound to a protein are pulled out and sequenced. This process happens again when the bound sequences are removed from their protein and then exposed again. The experiment results usually in 5 cycles, from cycle 0 until cycle 4. Cycle 0 represents the initial pool of DNA sequences, and cycle 4 represents sequences which were bound 4 times in a row. The next data set is protein binding microarray, or just PBM. It's an in vitro technology which is capable to produce roughly 40,000 sequences in each experiment. Those sequences are high resolution and represent all possible telomeres. The experiment starts by exposing transcription sequences to a transcription factor protein. Then the binding intensity of each sequence is quantified into a number, as you can see on the right. So each sequence has a number representing its binding intensity to a specific protein. The third data set is chromatin immunoprecipitation followed by sequencing, or just in short, ChIP-seq. It's an in vivo technology which is capable to produce low resolution subsequences. The binding occurs naturally in the cell since it's an in vivo technology. Bound DNA is sequenced and mapped to the genome. The experiments result in subsequences which are either bound or unbound. Since it's an in vivo technology, it suffers from several disadvantages. The first one is that it's noisy since there are secondary effects happening in the living cell, and it has low resolution since every subsequence is several hundred nucleotides in length. Artificial intelligence brought the bioinformatics significant breakthroughs. Artificial neural network is just a subclass of artificial intelligence algorithms. Convolutional neural network, or just CNNs, are a subclass of artificial neural network and can detect local features using dimensionality reduction. We can use convolutional neural network to predict protein to DNA binding sites. This can happen by converting each DNA sequence into a one-hot encoded matrix and then detect its local features using the convolutional kernels, as you can see down below. The computational challenge is to infer accurate sequence-based binding model from HT-SELEX data. This means to learn the binding sites from the HT-SELEX data. The important thing is to predict binding site on other datasets. If we learn and predict binding site on the same dataset, we cannot be sure that the model did not just learn a specific experimental bias. So the problem starts with the training data, which is HT-SELEX. This is the input to the deep learning model, which is the DeepSELEX model. DeepSELEX need to predict binding site that he learned from HT-SELEX data on 2 different data types. The first one is the PBM data, which is an in vitro technology. And the second one is the ChIP-seq data, which is an in vivo technology. So let's understand what DeepSELEX is all about. There are several HT-SELEX based algorithms. The first one is DeepBind. This is a deep learning based method to predict DNA bindings. It laid the foundation to predicting binding sites using deep learning. It uses synthetic negative as background sequences by randomly shuffling sequences. The second method is BSEM that predicts binding intensity using prediction weight matrices, or just PWMs. It is a probabilistic method that because its output are just PWMs. The third method is BindSpec that decode transcription factor proteins binding by sequence embedding. Its main purpose is to classify a sequence to a specific transcription factor. All of those methods treat the problem as binary classification problem, as the sequence has 2 states. It's either unbound or bound. On the other hand, DeepSELEX works quite differently by multi-label classification. The input of the model is a DNA sequence. The DNA sequence is converted into a one-hot encoded matrix. The second layer is a one-dimensional convolution layer with 512 kernel with width of 8 and stride of 1. And then we apply a max pooling operation, pool size of 5 and pool stride of 5. Afterwards, we have 3 fully connected layers with an activation function of value. The length of the fully connected layer are 64, 32, and 32. The output layer has a sigmoid activation function and an output shape of 5. Each output corresponds to a different enrichment cycle in the HT-SELEX data. Since each HT-SELEX experiment has its own DeepSELEX model, the input to the model is the length of the HT-SELEX experiment. Since the PBM experiments and ChIP-seq experiments has a varied sequence length, we need to apply a sliding window approach to enter the sequences into the model. Here, we can see that each sliding window outputs 5 results, 1 for each enrichment cycles. So if we have 3 sliding windows, we will have 15 results. Those 15 results need to be quantified into a single number that represents the binding intensity. This is done by an aggregation function, which is of the following form. We apply max of cycle 3, then edit max of cycle 4, as you can see, then subtract the min of cycle 0. This is all quantified into a single result, which is the binding intensity of the sequence on the left. How did we come up with this function? We first inspected how different function correlates to the actual PBM binding intensity. We found out that if we apply the max function on the higher cycles, such as cycle 3 and cycle 4, and apply a min function on the lower cycles, such as cycle 0, which represents the initial DNA pool, and cycle 1, we can have high correlation to the actual PBM binding. Then we need to integrate those functions to achieve a very good aggregation function. This is the best aggregation function that we found was to add the max of cycle 3, the max of cycle 4, and then subtract the min of cycle 0. As it turns out, fusing different enrichment cycles complement each other since they comprise of different type of information. Let's see some of the results. The first data set is the PBM dataset, which is an in vitro dataset. The number of transcription factor is 130. There's 139 HT-Select experiments, 214 PBM experiments, which result in 243 experimental pairs. Deep selects outperform its competitors by a large margin by achieving an average Pearson correlation of 0.416. The top competitor on this dataset is BSEM by achieving 0.362 average Pearson correlation and a p-value against deep selects of 1.2210 in the -4. This shows deep selects great statistical improvement. The second dataset that we inspected is the in vivo dataset, which is comprised from ChIP-seq experiment. It has a 32 transcription factor, which in 37 HT-Select experiment, 963 ChIP-seq experiment, which results in 1,097 experimental pair. We can see that most of the competitors achieve similar results when deep selects achieve 0.697 average area under the curve, and deep bind achieve 0.699. And the p-value between them is 0.83. We can see that this does not show any statistical improvement. And this is because, as we talked before, this is an in vivo data set, and some of the experiments are deeply damaged, as we show on the original paper. To visualize DNA to transcription factor binding site is extremely important, as we want to verify that the deep selects model did not just perform successful guessing on the sequences. Therefore, we generated logo by the top sequence on cycle 4 by the integrated gradient method. On the right, we can see several logos. The left column is logos from cis-BP reservoir for different protein, and on the right, you can see logos of the deep selects managed to produce. Those logos are very similar. This shows that deep selects learned informative motifs. New binding sites can help us to understand gene expression mechanism, which is still under research. Several conclusions. Multi-X classification provide better predictive power as the sequence are better separated between their states. Aggregating several enrichment cycles utilizes all of the experiments information, since different cycles comprise of different information. Cycle 0 is preferred to mimic background sequences over random generating, because this way we can keep the statistical properties of the initial DNA pool. For future work, we plan to improve the aggregation function, which always use 3 fixed enrichment cycles, which are not always the best for each HT-Select experiment. And we plan to use TF-Modisco to summarize different sequences to produce logos. I want to thank my instructor, Professor Yaron Orenstein, which gave a key insight to the development of deep selects, and to Intel, which contributed us the AI Dev Cloud, which helped us produce a deep learning framework. Thank you very much.
Thank you, Maor. Any questions from the audience? No, not for the moment. So how good are you in linking a motif with a transcription factor?
Very good. We show it on the final on the original paper, and we want to improve this by using the TF-Modisco model, since currently we are generating logo by the integrated gradient descent, which in this way the logos are generated only by one sequence, which is the top sequence on cycle 4. By using the TF-Modisco, we plan to summarize the different logos to produce better binding sites. And we showed that by predicting on in vitro datasets such as PBM, which is in high resolution, we are giving very good results, especially by using the output of cycle 0, which gives a very good separation between which is unbound to the other cycle, which might represent a more probability for a sequence to bind.
Okay, thank you. And just to save some time, we'll get to the next speaker. So, which is, let me check to be sure. Emilio Dorigatti from the Ludwig Maximilian University of Munich. So he will be talking about epitope-based vaccine design. So the floor is yours, Emilio.
Thank you, Stefan, for the introduction. I'm very excited to be presenting my work here. I'm not sure I can change slides though.
You should be able, yeah.
Okay. Oh, okay, it's not optimal.
It's a bit slow.
Well, when you design a vaccine, in general, you start with the antigens that are the bad guys you want to fight with the vaccine. And you analyze their genome and you extract epitopes that are short fragments that can be recognized by the immune system. And then you need to select a few of these epitopes to be inserted into the vaccine, and you stitch them together using short sequences that are called spacers that are put between the epitopes to link them together. And in this work, we propose a new framework to do this step that simultaneously selects the epitopes to be included in the vaccine and assembles them. And this is unlike other state-of-the-art methods that only focus on one of these steps. When the vaccine is injected into the body, it will be eventually digested by this cylindrical shaped protein called proteasome. The proteasome will cut the vaccine into several pieces. And for the vaccine to be effective, it is important that these pieces are exactly the epitopes we inserted in the first place. And we use stochastic simulations of this cleavage process. And we show that with our method, we can increase the number of epitopes that are recovered from the vaccine, which means that eventually the vaccine is more effective. Before going into the details, I would like to talk a bit about variability. So mutations are in general one of the most important ways that pathogens use to escape the immune system. However, not every part of the pathogen can mutate. So what you see here, you see a portion of the HIV genome, and for every position, we calculated the entropy, which indicates how frequently this position mutates. And as you can see, while some positions are essentially random, others are very conserved. In particular, these three regions are known to be extremely important for the functioning of HIV, so that if they are mutated in the wrong way, the resulting virion will not be effective. And so by natural selection, these regions will be conserved. The HLA is a protein complex in our body that presents antigens to the immune system. And this step is extremely important for the immune system to recognize and fight the pathogens. And there are thousands of known HLA alleles, and their geographic distribution is highly variable. And the key idea is that the variability in the antigens is translated into variability in our immune system. And we need to take care of this when we design a vaccine. Now, our framework is based on linear programming. The objective function is to select a set of K epitopes that together have the largest immunogenicity, which is quantified as the average binding affinity to the HLA molecules. On this, we add constraints for the cleavage score at the terminals of the epitopes. So the cleavage score is proportional to the likelihood that the vaccine will be cleaved in that position. And we want the cleavage at the terminals of the epitopes to be very high so that the proteasome, when digests the vaccine, will separate the epitope from the rest of the vaccine. We also add a technical constraint bounding the cleavage score inside the spacers. Then we have a constraint that upper bounds the cleavage score inside the epitope itself, because we do not want the epitope to be cleaved in the middle, but we want it not to be preserved. And if it's cleaved, then the vaccine is not effective. We have then constraints that force the linear program to select epitopes that have a minimum coverage of antigens and a minimum conservation. This means that the vaccine will target regions that do not mutate very frequently and is able to cover a large part of the strains in circulation. And finally, we also force the linear program to select epitopes that cover a large portion of HLA alleles so that the vaccine itself can be effective against a broad population. To go into more details about the structure of the linear program, we have binary decision variables for every epitope in every position and for every amino acid in every position of every spacer. In this example, we have a vaccine with 3 epitopes and 2 spacers with 4 amino acids each. Any given choice of these decision variables will give us a specific vaccine with certain epitopes in certain positions and a spacer constructed in a certain way. Given these decision variables, we can reconstruct the sequence of the amino acid, the sequence of the vaccine. And then based on the sequence, we use a position-specific scoring matrix to find the cleavage contribution. So how much an amino acid in a position P will affect the amino acid in a position Q. And notice that since our spacers can contain gaps, for example here, then we need to dynamically compute what is called offset, so the distance between P and Q. Once we have these cleavage contributions, we can sum them to obtain the final cleavage score for every position. And given our choice of epitopes, we can finally compute the antigen coverage and the epitope conservation. And these quantities computed in steps 4 and 5 will be the quantities that are constrained in the linear program. So the cleavage score is actually a linear model that gives us the log odds of cleavage conditioned on the 4 preceding amino acids and relative to a certain prior cleavage probability. Since we want to compute the unconditional cleavage probability, which means that we first have to fix a given prior, and then in order to remove the conditional, we define a stochastic process which forbids cleavage if the cleavage happened in the 4 positions before. Otherwise, if this event did not happen, then cleavage happens with the probability given by the linear model. We take several samples from this stochastic process, in this example 30, and you can see the gray boxes indicate that cleavage happened in a certain position. Now, since we are interested in the epitopes, consider this epitope here, for example, we say that this epitope is recovered from the vaccine if it is cleaved at the boundaries and not inside. And this, for example, happened in these 4 simulations. So cleavage happened here and here, which are the terminals, and not here, which is inside. And so we estimate the recovery probability of this epitope to be 4/30 or 13%. And finally, we use these recovery probabilities to compute the expected value of certain metrics. And this expected value is what we use to evaluate the vaccine. Now, in this slide, you see two vaccines that were designed with a sequential design and with our solution. And for every position in the vaccine, you see the cleavage probability that we computed using the simulations. The green bars are the terminals of the epitopes, which is where we want cleavage to happen, and orange is where we want cleavage not to happen. And sequential design means that we use state-of-the-art tools to select a set of epitopes that has the largest immunogenicity, and then we use the second tool to find the best way to assemble them. So the epitopes selected by this sequential approach have a much larger immunogenicity than what can be selected with our approach. But this happened on purpose because with our solution, the selected epitopes have a much smaller cleavage probability inside. And this means that we can greatly improve the recovery probability of the epitopes. In fact, out of 5 epitopes on average, more than 3 are recovered compared to less than 1 for the sequential design. And this increased recovery rate translates to improved immunogenicity of the whole vaccine. So these expected immunogenicity and the cleavage simulations are computed only after the linear program is solved. And so if we want to maximize this, we need an outer optimization loop, for example, a grid search. So in this case, we are running a grid search, changing the maximum score we allow inside the epitopes and the minimum score we want at the terminals. And for each combination, we compute the expected immunogenicity. And we repeat this process for several cleavage probabilities, prior cleavage probabilities. And what you can see is first, the best results seem to be obtained with a prior of 10%. And second, as the cleavage probability, as the prior increases, we need to be more strict with the maximum score inside the epitopes, and we can be more lenient with the score at the terminals. So this prior probability will also, of course, influence the number of epitopes that are recovered. And here we quantify this number for a wide range of priors for our solution, which is the blue line, and for the sequential approach, which is the orange. And it's clear that with our framework, we can greatly increase the number of epitopes that are recovered across all range of prior probabilities. And in turn, larger number of recovered probabilities of recovered epitopes translates to better metrics. So higher effective immunogenicity, higher coverage of antigens, and higher HLA coverage. So in conclusion, in our conclusion, even though the state-of-the-art methods tackle these two problems of selecting epitopes and assemble them separately, with our framework, we show that it's possible to do it at the same time. And this results in more effective vaccines for the very simple reason that more epitopes are recovered. And this is because we can influence the selection of epitopes based on the cleavage pattern. So I would like to thank you for your attention. I would also like to thank the Munich School for Data Science, which is my graduate school, and my supervisors, Dr. Benjamin Schubert and Professor Bernd Bishop.
Thank you, Emilio. Any questions from the audience? No. Have you ever been able to test one of your designed vaccines in vivo?
Unfortunately, no. And we would love to do this. Actually, since we are a small lab, we don't have a lot of collaborators. So if you're interested in using this framework, by all means, please write me. It would be very nice to test it on.
You didn't contact anybody working on vaccines?
Yes, we are starting to find contacts, but the lab is very young, and so we are still building our network.
Okay, no more questions. This is not. Okay, we're moving on pretty well. So I don't see any more questions? Okay, so I guess we'll move on with the next one. Thank you very much, Emilio. So next one is Adelme Bazin. I don't know if I pronounce your name correctly, sorry. Bazin, probably, from Genomes in Évry in France. And he will present a method to predict genomic islands in a pan-genome-based environment. Adelme, the floor is yours.
Thank you. Thank you for the introduction. So hello, I am a second-year PhD student working at LabGen Genoscope CEA in France. And I will present to you RGP, which is a method to detect genomic islands in microbial genomes. So nowadays, microbial genomic studies rely on the comparison of thousands of genomes in a species. Those genomes display a very high diversity in terms of gene content that comes from horizontal gene transfers. Between 5 to 40% of genes are variable, and they also display a very high level of polymorphism. So this gave rise to the concept of pangenomes. Pangenomes is the union of sequence entities shared by genomes of interest, usually from a given species. And it gave rise to new data models that move from capturing only the specificities of a single genome to the overall diversity of a reference pangenome. So pangenomes can be worked on at the sequence level, like you may have seen on trends in bioinformatics on Friday, but also it is worked and originally worked on at the gene level. So the goal is to study the diversity of genes shared by a group of individuals. A pangenome is built as such. First, genomes are annotated, then they are clustered into either homologous or orthologous gene families, and then they are partitioned into either core or accessory genome. The core genomes are genes that are present in all genomes of the species, and the accessory genomes are genes that are present in either one or multiple genomes of a given species. So while this classification is very straightforward, it is not very practical, since nowadays a lot of genomes that we work with are fragmented. So in 2008, Kunin and Wolf came up with another way to classify, to partition pangenomes. That is the relaxed core or persistent genomes that includes genes that are present in most individuals, but not all. The shell genome, which are genes that are present at intermediate frequencies, and the cloud genome, which are rare genes. A lot of tools were built to partition pangenomes into 2 or 3 classes, as I just introduced, but most of them are based on the frequency of gene families, and they are hard-coded thresholds. Recently, a tool from our lab called Pangolin was published. It is based on a graph structure, as you can see on this slide, and a statistical partitioning algorithm called neighborhood expectation maximization that uses both the presence-absence of gene families and the graph structure to partition the graph. The method to detect genomic islands is based on this graph. So here is an actual graph from a pangenome built from 3,000 genomes of Acinetobacter baumannii. So the orange stretches are persistent genes, and the blue and green are shell or cloud genes. So you can see that there are stretches of orange separated by multiple paths of green or cloud, of shell or cloud genes. Those paths are called genomic islands. So genomic islands are specific areas of microbial genomes. They're often associated to tRNA genes, integrase, or insertion sequences, and they aggregate strain-specific adaptations, such as environmental adaptation, pathogenicity islands, antibiotic resistance mechanisms, or secondary metabolites. They display a very high turnover in terms of gene content, and they often show composition bias. Because of this, in the scope of pangenomics, they are expected to have mostly shell or clad genes. So we decided to try to detect those genomic islands by finding regions with a high number of shell or clad genes. So the method goes as follows. We annotate genomes, we cluster genes into homologous gene families, and then we build the neighborhood graph of the pangenome. Then we partition it with pangolin. Then we project the partitions onto the genomes in place of the genes. Then we compute a score using the formulas that you can see along the contigs. The high scoring regions will be those that are made of shell or clad genes. We benchmarked the method against two reference datasets that were published, first in 2008 and then updated recently in a 2018 paper. The first dataset is called the comparative dataset. Is built from comparative genomics and filtered down to 81 genomes. In this dataset, MyRGP comes on top of the 10 other tools we tested it against, very close to Island Viewer 4, which is a state-of-the-art tool. The second dataset is much smaller, but also much more reliable as it relies on published descriptions of genomic islands. It is made of 6 genomes. Here again, our method comes on top of the other method we compared it against. So what we detect is called regions of genomic plasticity, and we did not specifically name it genomic islands for a few reasons. So as you saw, genomic islands are well detected, as you saw with the benchmark, but regions of genomic plasticity can be a little different. First, they can correspond to both gains or losses of genomic regions. That is, if some genomic regions are lost differentially throughout evolution, there will be regions of genomic plasticity, but they are not genomic islands. RGPs can also be plasmids. And some genomic islands can be persistent within a species, or sometimes the classification relies on functional types rather than just the presence in a plant genome. So this is a circular representation of a Salmonella enterica CT18 genome. The outer layers are the genes. The inner layers are genomic island predictions. The one in red is PanRGP, and the one in black are the literature genomic islands. So PanRGP globally detects relatively well the literature genomic island. I'd like to zoom on one in particular. So this is a genomic island that's not detected by any tool. This is because it's mainly persistent. It was classified as genomic island because of its functional type. So as I said, genomic islands display a very high level of gene repertoire changes, and most gene repertoire changes are gathered into very few hotspots. So in this picture, you can see multiple genomic islands in a single hotspot. So the colored squares are groups of genes, and there are sometimes no similitudes between the genomic islands. We define spots of insertion by the persistent genes that flank the genomic regions rather than the gene content because of this. And genomic islands of the same spot can have unrelated gene content. So we designed an algorithm to detect and group genomic islands into spots of insertion to study their evolution throughout the species. The algorithm goes as follows. We rely on the flanking persistent genes families rather than the genomic islands themselves. We build a graph where each node is a genomic island, and we compare the borders of different genomic islands. If the two borders are similar or if they overlap, we add an edge in the graph. The order in which they appear does not matter. We add multiple edges to the graph. If only one border matches, we do not add an edge. In the end, the connected components in the graph are the spots of insertion. So as there are no reference data sets for spots of insertion, we just looked for the one we knew, which is the LEX hotspot in E. coli that I showed to you earlier. We built a pangenome from 1,400 E. coli metagenome assembled genomes from a recent publication and looked for the hotspot using the flanking persistent genes. The hotspot is represented here as a pangenome graph, where you can see in orange the flanking genes and the multiple paths that are the multiple genomic islands that are in the hotspot. The stay-home message for this presentation is that RGP uses a pangenome graph to accurately detect genomic islands and spots of insertion. Studying genomic islands in fragmented or incomplete genomes like MAGs is possible. And PAR-RGP can be used through the Pangolin framework or through the microscope platform. The Pangolin is installable through GitHub or Bioconda, and microscope is available at this URL. Thank you to the people that participated in this project, and thank you for listening.
Thank you, Adelme. Any questions from the audience? Nothing yet. We still have some time. We still have lots of time. So if there's any questions for the other presenters, maybe we can still fill up a little bit of the time. But I have a question for you, Adelme. Did you try with larger genomes? Because now you presented only bacterial genomes.
Yes, I did.
And they are definitely more simple and smaller. How does the system scales?
So in terms of scaling, we managed to compute genomic islands on over 15,000 genomes at once on microbial genomes, that is. We did not test it on eukaryotic genomes, but if someone is interested to do it, I'll be happy to know.
Okay. I have a question from the audience. What do you see a certain bias in the function of the genes in such dynamic islands across different species? Or
yes, so we expect to find genes often associated to selfish genomic elements, for example, like phages. And there are also very often specific functional types, such as environmental adaptation, because those genes are often shared by horizontal gene transfer between bacteria that live in the same biomes.
Okay. I have a question for Emilio still. What about MHC class 2 predictions?
Sure. So our framework is in general agnostic to the immunogenicity function that you choose. And you can, in principle, you can combine MHC class 1 and class 2 epitopes as well as B cell epitopes, although it's not implemented yet, but it's certainly possible given the linear programming formulation.
Thank you. Another question for Adelme. Now that you mentioned MAGs, the identification of genomic islands could be used to differentiate strains in a single MAG.
Yes, so that's a very good question. So if we know some genomic islands are associated to specific strains, as it is often the case in E. coli, for example, then yes, definitely we could, if we find a genomic island in a MAG, we could definitely associate it to a given strain. But if the genomic island is uncharacterized previously, we wouldn't be able to associate it with a strain, I guess. But yes, it's definitely possible.
Mm-hmm. Maybe I have still a question for you. Suppose you have let's say a population of a certain bacteria, mm-hmm. and within that whole population there is some strains that have some pathogenicity, yeah. But this pathogenicity is sort of underrepresented in the whole population and mm-hmm. only emerges once in a while as a sort of an outbreak.
Yeah.
Would you be able to sort of identify those?
So yes, because since it's only present in very few individuals, the genes that are responsible for the pathogenicity will be classified as shell or cloud by the pangenome method. So if they gathered in a region where there are more than few shell or cloud genes, which is usually the case, we'll definitely be able to detect them. The only case in which we would not be able to detect the pathogenicity genes is if the whole population has those genes. In that case, we would not be able to detect them. But usually it's not the case. So yes.
Should be fine.
Should be fine.
Okay. I have a question for Maor. How does your method compare in memory and running time compared to the competitors?
Actually, very swiftly. We did not compare our method to the competitors, but I can say that training one deep learning model per HT cells experiment is a matter of a couple of minutes, because as we show in the article, we use only 15,000 sequences from each enrichment cycle. BSEM, which uses all of the data, which takes roughly 40 minutes to an hour to train one model. We can train one model in 3 or 4 minutes, and training for all 214 HT-Select experiments on several computing nodes takes less than half an hour. So it's very swiftly. If you just want to check our model on a specific HT-Select dataset, you can build up an environment and have all the models in less than an hour. We also have a very well-documented model on GitHub if you want to take a look.
Thank you. Any more questions from the audience? Doesn't look like. Okay, we still have a couple of minutes. But if there is nobody with emerging questions, I guess we can close the session. So I would like to thank everyone, all the speakers, for their very swift presentation, clear and on time, more than on time even, which is surprising. Thank you very much. Thank you for your presentations. And I guess that the speakers are somehow contactable still through different chat systems. If there would be any more questions that people would like to discuss in more face-to-face or in a more direct chat. Thank you to the speakers again. Thank you to the organizer for setting up this whole system. And I hope to sometime meet you live, maybe, in the future. Thank you.
Thank you very much, Stefan.
Thank you.
Thank you.
Bye.
Thank you.
Bye-bye.
Bye-bye.
Bye.